Before I start my presentation, I would like to say that my presentation, that my work is more suitable and more beneficial for the youngsters and young students, but I am not getting any in this room.
You're calling us old? That's our job.
Youngsters, I mean to say youngsters. I have worked on Antarctica. I am fortunate enough to be there for 2 times. Actually, in Antarctica, it's a no man's land. No country claims on Antarctica. And several countries, more than 20 countries, have made their stations over there in Antarctica. They are going there, they are for the scientific purposes only. They go there, they work, they found the results for the benefit of mankind and the science and society, and they come back. Likewise, our country also manages to send a scientific expedition every year. And I am fortunate enough that I have been deputed by Government of India twice to work in the field. And I have done my PhD and postdoc and everything on Antarctic microorganisms. I have worked on diversity, physiology, morphology, biochemistry of the microbial plants. But this presentation is on algal diversity of freshwater streams of Schirmacher Oasis, Antarctica. Antarctica is situated on the southern part of the globe, and Antarctic flora has evolved millions of years ago on the supercontinent of Gondwana land. And the separation of South America from Antarctica 30 to 35 million years ago allowed the Antarctic circumpolar current to form. It's a stormiest, windiest, coldest, and least accessible continent. This is the map of Antarctica, and as I said that more than 20 countries have made their stations over there, and the US is the pioneer in establishing the station over there and working in this hostile environment. US has this Amundsen and Scott station. Amundsen and Scott were the 2 scientists who went there for the first time. And India has established a station Maitri, Priyadarshini, after the name of our late Prime Minister Indira Gandhi. So in Antarctica, we don't have any plant. We don't have any berries, fruits, flower, root. We have only 2 grasses, and that too on this peninsular region. No plant is there. Now, this is the Schirmacher Oasis. This is the region, actually 2% of whole Antarctica is ice-free region. And the India has established a Maitri station in the Schirmacher Oasis region. This is the map of Schirmacher Oasis region. This is the Russian station, Novolodorevskaya. And nearby this place, George of German station is there. This is the picture which shows how the algae and only the microplants are growing there. For example, algae, fungi, bacteria, and some fauna is also there. So this is the picture which shows that this is the permanent glacier, and when it melts, the water flows like this. And in between these places, the smaller plants like bacteria and algae used to grow. Again, these are the pebbles and stones, and the microalgae and fungi and bacteria, they used to grow in between the two pebbles or two stones, just to avoid the the cooler winds, and just to avoid the 6 months day and 6 months night period. We have 6 months day and 6 months night in the Antarctic itself. Now the material and methods, we have calculated the alpha diversity, richness, species richness, evenness and equitability, beta diversity, gamma diversity, cluster analysis of the various algal flora we found over there. These are the various algae which we found over there, and the algal and cyanobacterial species which have been encountered in different streams are enumerated in this table. It is evident that various streams situated at different locations varied in flora composition. Just because of the presence of the rookeries of the penguins. The penguins do occur in hundreds and thousands in numbers, and their excreta enriches the environment by the manures and all. The glacier-bound streams near Maitri Lake shows maximum abundance of cyanobacteria, which is the number of, whereas the number of species encountered was minimum in hilltop snowdrift bound streams near German station, just because the availability of the nutrient. Among the cyanobacteria, more than half of species were nitrogen fixers. And oscillatoria, formedium, and some other species, they are found in abundance. These are various algae we found over there: Crucoccus, Cosmarium, Glyocapsa, Lingvia, Oscillatoria, Formedium, Chemisiphon, Plactonema, and nostoc. I have worked more specifically on nostoc. My PhD work is on nostoc species, and it's a nitrogen fixer, and it fixes nitrogen even when, as we know, that our metabolism works at a particular temperature. Likewise, the plant also, the metabolism of plants also work at a particular temperature. So, in Antarctica, when we experience minus 40, minus 30, minus 20 degrees centigrade, it's very difficult for these smaller microscopic plants to sustain themselves and to grow, divide, reproduce themselves. But even though these microplants grow over there, in the open atmosphere. Now the result is we have calculated the Simpson index, Shannon-Wiener index, Manning index, various species evenness and richness, etc. We have illustrated the results with the help of cluster analysis depending on the similarity of the lakes we have observed. These are the various photographs in which we can see these are the places which are ice-free, and that too for the 2 or 3 months only. After that, they are again covered by thick snow. And this is the vessel which take us to that very place. These are the choppers which we take us to the various sampling sites and experiment site. This is the Maitri station over Antarctica, and this is the huge rookery of the penguins. Now, I would like to acknowledge the Department of Botany, Banaras Hindu University, and for their laboratory facilities, the Department of Ocean Development, who gave me the opportunity to participate in this in the expedition twice, and the Professor A.K. Kashyap, my supervisor, and my team members for working on the Antarctica and Antarctic diversity, Antarctic fields, Antarctic physiology, and all. That's all. I would love to answer any question.
Yes, we do have time for questions. The floor is open. Sir? Yeah.
The isolated
so many algae.
That to the Antarctic, because that is what the beauty of the microorganism. Because that
Yeah,
is the
they
one that can
used to grow everywhere,
Yeah.
cosmopolitan.
So I'm
From
s-
that you can expect any novel metabolites. From that you can cure any disease like that.
Actually, I started work on, first of all, I started work on how
if I
they...
could interrupt, I'm not sure everybody can hear him. I don't know where the microphone.
Microphone.
Just go right ahead.
Because that is the one, it is unique, because
Yeah.
the beauty of that microbes is survives everywhere.
Yeah.
In minus 60, minus 40, we don't know the mechanism. From that it produces any metabolites that could be useful for as a new drug invention, like it may be anti-cancerous or other things. That is what my question. If you tried
algae,
that's
normally algae do have so many secondary metabolites, but in Antarctic algae, first of all, we started to work on what are the conditions, how they establish themselves. First motto was that how they establish themselves in such a hostile environment. For that, I started working on plasmids. And I got success in isolating plasmids in Nostoc commune. And, but I couldn't work on metabolites, which are, yeah.
Other questions? No? Oh, let's give...
Since the conference was on diversity, I restricted my work on a small part of diversity only. Otherwise, I have worked on nitrogen fixation. And my PhD work is on nitrogen fixation by Antarctic cyanobacteria Nostoc commune. And
But that's very good part of biodiversity.
What is?
I say that's a very strong factor in biodiversity. That
Yeah.
those can survive in such harsh environment.
Actually, they have the adaptations.
Yes.
They have the adaptations.
Yes.
The seal which found over there, they have a very thick blubber in their body. The penguins, they have a very strong this thing, the thick body, and these are their adaptations. And in my algae, they do also have their adaptations. Normally, when we find algae in our area, in the tropical area, we don't have any covering on that unicellular algae. On any we don't get any covering, but they do have a cover on them. They do have a cover on them. They used to grow in clumps, not they grow separately, just to avoid the winds, cooler winds and all. So they do have some adaptations, but scientifically what we can say that they do, actually I could find that they have a plasmid. So it might be a possible reason that they are surviving in such a hostile environment.
Dr. Rajan,
Sir.
you are from Himalayan state,
Sir.
and in Himalaya also you can get such kind of
Right.
adverse condition with
Right.
minus 40-degree
Right,
temperature.
right.
Do you ever tried that to get any algae from those regions of Himalayas similar to having the temperature of Antarctic and the windy area and something like that?
Actually, this thing, the factors are even the Himalayas are cooler, and Himalayas we do have ice and such type of things, but the conditions are entirely different. Conditions are entirely different.
I'm interested to know if you tried any time at the higher altitude in the Himalayas to find out if there
I do have collected the similar algae from there also, from the Oli, from the Gomukh, and from the other regions also, but they are not similar to those algae, those nostoc or those algae which are occurring over there.
Also, it might be of interest to the group that NASA has astrobiologists looking at what they call extremophiles, which is exactly what Rajan's talking about, and they're in the Death Valley, Antarctica, underwater vents. So yeah, they're looking all over.
I invite all of you to work in the Antarctic field, in the specific area you are working on. The people, our country used to send the geologists, the zoologist, the botanical survey person, and meteorological department scientist, and climatologist. All those scientists used to go. Even with me also, there are so many scientists. They accompanied me over there, and we have worked together, and we have got a very good results. We used to search the possibilities of growing higher plants in open. Of course, we couldn't get success in that, but we tried a lot. We prepared a greenhouse over there. We prepared a greenhouse over there, and we maintained the temperature. We grew some plants like chili and such type of plants in hydroponics using the biofertilizers. That's what I was in the morning session saying, that why can't we use biofertilizer while we are using biofertilizers in such a hostile environment. So